My name is Dr. Richard Cripps. I'm a professor in the Department of Biology at the University of New Mexico. So the study is to try and understand how different muscles are formed inside of the body. And so we were fortunate in one of the experiments that we were doing to observe an interesting effect when we changed the function of one particular gene. It actually turned one muscle type into another muscle type, essentially a slow muscle into a fast muscle. So we choose fruit flies for this project because they also have muscles that form a variety of functions inside of the body, and the genes that are involved in creating those muscles are very similar to the genes that work in human bodies to create muscles as well. So by understanding genetic processes controlling muscle formation in fruit flies, we can gain basic insight into how those same genetic processes might work to control muscle development in humans.
Muscle development is very well organized, and certain players come on and off during certain situations. And so I look at which players turn something on to result in proper formation or improper formation. And so what I do is I take out those players, and then I see, does the fly have a phenotype? And what we mean by phenotype is it is unable to fly, or it's unable to jump, or it's unable to do both. Our focus is to apply Drosophila to muscle diseases, and ours is muscular dystrophy.
So we've identified some of the players that control that regulatory process. What we really need to do now is to find out what other genes these factors regulate, and in turn perhaps what regulates them as well. So we're somewhere in the middle of this important and interesting, we think, regulatory network, and we're really trying to find out what the separate players of that process.
So we've come up with this specific approach that could allow us to track the development of different muscles in the fly body. So it took us quite a while to develop it and get it running, but then in the end we found that under some conditions we can turn one type of muscles into another type, and that's what we published.
So publication is a tangible indication of having made progress on the project, and that's important because funding agencies such as the NIH and the Muscular Dystrophy Association fund our research, and they want to see that we are making progress on the work that we've done.
I was able to see it from the beginning because it was from my screen that we saw these transcription factors were important for muscle identity. And so to see something progress and you find new things out and you discover, other key players in this process, and then you see it culminate into a publication, I mean, that's amazing because you're like, wow, my hard work went to something. And for me, it's important because it's my first publication, and so I'm really excited about it.
We're just, having fun, just trying to understand how things work in the world, and that's what makes us very excited.